Good morning, everyone. It's really great to see so many of you after the nice celebration we've had yesterday, and you made it still into the morning session. I think we will slowly start, not lose any more time. It's my great pleasure to introduce our keynote speaker of the first session today, Anders Krogh. Anders Krogh is a professor at the University of Copenhagen. He is, in his work, focusing on medical data analysis and machine learning these days. My personal first contact with him and his work, though, was much earlier, actually, when I just started my journey in bioinformatics as a student, where we had to study his amazing book on biological sequence analysis and hidden Markov models, and also his work in that direction, like TMHMM, was a major thing at the time and also very important for my own work. So for that reason, I'm really delighted to be able to welcome you here and look very much forward to your talk. Thank you for joining us.
Thank you very much. Can you hear me? Thanks for the nice introduction, and thanks for inviting me to this wonderful conference, although I'm not part of this cozy bioconductor community. I've been doing bioinformatics all my career, but lately I've been switching more towards deep learning, where we're using Python and not so much R. So I'm going to tell you about a deep generative model we developed that we apply for gene expression and at the same time other data. That's the reason for the funny title, gene expression and multimodal data. So generative models are really hot these days, of course, because of generative AI driving the ChatGPT and so on. I'm going to talk about sort of a very simple type of generative model where you assume that your data live in a high-dimensional space, but they are very structured. So you think of them as being on manifolds in this high-dimensional space. So in this cartoon here, we think of images living in these structured objects in high-dimensional space. And the objective of latent variable models is to try to make a low-dimensional space that reflects these high-dimensional structures. It's very much like PCA. PCA is a latent variable model. Typically, you have a 2D PCA representing your high-dimensional data. In PCA, you have a linear mapping from the low-dimensional space to the high-dimensional space. In these more sophisticated deep models, we have a deep neural network mapping from the latent space to feature space. So F is implemented using a neural network. But the idea is that for each point, for each sample in feature space, we have a point in this latent space, and this point maps into the sample. Then to make it a generative model, we need a probability distribution on the latent space. Typically, we use a simple Gaussian, because then we can sample from the distribution. Once we sample a point, we can project it into feature space, and we have an image or whatever we're modeling. So this is how image generation models work. You probably, a lot of you have heard about the variational autoencoder, and many maybe even use it. And this is an example of such a generative model. So in this model, you have the latent space, and you have a decoder implementing this function that maps from latent space to feature space. The trick in the variational autoencoder is to train the model. They introduce an encoder that maps your sample into latent space, and then the decoder maps back into feature space. So it's very easy to train an autoencoder because you train it to reconstruct the input. To make it a generative model, you need to add some tricks, and then the math becomes more difficult. So you have to do variational inference. We're not going into the math at all here, but in the VAE, you maximize something called the variational lower bound, the evidence lower bound. Despite the elegant math, this is still an approximation. Sometimes it's actually a pretty bad approximation, especially if you have small sample sizes. So we have come up with an alternative model where we don't have an encoder. This is somehow very slow. So instead, we still have the same setup with the latent space and the decoder mapping from the latent space to feature space. But here we treat the representations as we treat parameters. So it means that we are optimizing the representation for each sample in the training set at the same time as we are optimizing the parameters of the neural network. So we start training, you know, when we train neural networks, we start with random weights and biases in the neural network. Similarly here, we start with a random point in latent space for each training sample. And then we maximize the likelihood of our data in both the latent representation and in the parameters of the neural network, all at the same time using standard gradient descent. For those of you working with neural networks, we just use the Atom optimizer. We add also a probability distribution over latent space. So in the variation autoencoder, typically you use a standard Gaussian fixed. Here we introduced a parameterized distribution to model clusters in latent space. Because a lot of the data we're working with, we expect clustering. For instance, for single cell data, you expect clusters corresponding to the different cell types. So we use a Gaussian mixture model, and we estimate the parameters of this model. So clustering is sort of an integral part of the model. So we know how to learn representations for the training set. These are just optimized together with the rest of the model. But what about new samples? When you don't have an encoder, we can't map directly a new sample in the model. So we actually have to find the most likely representation. So we actually optimize the same loss function as we do when we train, but we keep the model fixed. So all the parameters in the model are fixed, and then we just do gradient descent on the representation for this new sample. And this way we can deal with new samples and test samples. The magic of this is that it works also for missing data. So in the variational autoencoder, because of the encoder, if you have missing data in your sample, so if some of your data points are missing, which is extremely common when we model medical data, you have to impute them because the encoder wants a value. So you have to put a 0 or the mean value of your sample or something like that. But in the DGD, the deep generative decoder, we can simply leave them out of the likelihood because the log likelihood is a sum over samples and a sum over features. And if a feature is missing, we just leave it out. So we just sum over those that are present. So we can find the representation for the based on the features that are present. And then actually we can predict the missing values. We can do imputation because once we have the representation, when we push it through the decoder, it gives a value also for the missing values. So we can use the model in this predictive mode also. We can also leave out a whole bunch of variables and predict them from all the rest. And I will show you some examples of that. So I promised to talk about multimodal data. The first model I will talk about is a model we developed for attack-seq and RNA-seq. Gene expression and chromatin accessibility. And the model is as before. We have the latent space, the Gaussian mixture model, the decoder. The only difference is that we now have two sections of output. We have the gene expression, the RNA-seq data, single-cell RNA-seq, and we have the attack-seq data. So these are all counts that we model with a negative binomial distribution. These are pretty large, high-dimensional data sets. You can see here we tested the model on 3 different data sets, and you can see that there's quite a lot of features. So typically you have maybe 15,000 genes you want to model in the human. You can also model at the transcript level, which we do in one of the data sets. But then the attack is high-dimensional because typically you divide the attack-seq into peaks, roughly 100,000 peaks across the genome. You can see it varies here, but it means that the number of features we're modeling is between 100 and 200,000 in this model, with a fairly modest number of samples sometimes. So the first thing we did was to see how does this model fare with small data sets, because that's one of the advantages of this model, that we can model smaller data sets than a lot of other models. What we show here is the test loss, which is essentially the reconstruction error of the model, and we show it relative to using all the data. So when we use all the data, the ratio here is 1, because we compare these subsamples to the full data set. Then we look at 75% subsample, 50%, and so on. So this is a percentage of the training set we're using. And you can see for our model, the reconstruction error is pretty stable, down to 10% of the training set. Whereas we compare here to multi-VI, which is sort of a standard in the field, and you can see it doesn't do as well with small sample sizes. This is based on a variational autoencoder. We can also show that we have good reconstruction on these data sets, both on the attack and on the RNA-seq. Don't have time to go into a lot of details here. One of the things that fascinates me is how can we use these models to predict perturbations? They are trained on static samples. Do they learn anything about interactions between genes and chromatin structure, for instance? We can actually study that in this model by doing in silico knockdown, for instance, of a gene. So here we have this represents the mRNA-seq. This represents the attack-seq. We can put one of the outputs to 0, the output corresponding to a gene. And then we can calculate using gradient descent what happens to the representation. So we take one step of gradient descent. So this represents this one step. And then we can see if we have this representation, we get this output. If we have that representation, we get this output. So we can look at the difference. And we looked at the difference in the attack count. We call it delta X. And for instance, we can see that the main change in accessibility happens close to the promoter of the gene we knock out. Here we knocked out 2,000 highly varying genes, and we measured the average delta X in the vicinity of the gene. So you can see It's largest here near the transcription start site. These numbers are very small because we're taking a tiny step in latent space, but it shows a direction. If you keep following the gradients, quite often the model will get into unknown territory and output garbage. But for these small perturbations, we can actually see that it certainly learns interactions, because this makes a lot of sense that it's the core promoter that is affected. We also applied the model to microRNA. So in this example, we use the cancer genome atlas samples that have paired mRNA and microRNA. This is bulk data. So instead of modeling gene expression and attack, we here model gene expression and microRNA expression. Otherwise, it's essentially the same model. So the TCGA has these roughly 30 different cancers. This is just an example showing predictions for MIR21, where you have the true prediction across the different cancers. The dots are the samples. And the predicted expression of these microRNAs across here. And I forgot to say the most important. This is microRNA expression predicted from mRNA expression. So the objective in this project is to be able to say something about microRNA expression when you only have mRNA. And we also apply it now to single cell data, although as far as I understand, there's very little single cell where you have both of these modalities. I should also stress that this work is happening in Jakob Skov Peterson's group at Aarhus University in Denmark. So I'm not leading this study. The next example is truly multimodal, because here we are modeling gene expression data and clinical data together. So we have clinical data of many different types. For instance, we have survival. We have categorical data like treatment type that we want to model with a multinomial distribution. We have continuous data that we want to model with a normal distribution. We have count data, for instance, the mRNA data, where we're using a negative binomial. So the idea here is to make a model that where you can actually use the correct probability distribution for each of your variables. But we're still optimizing the total likelihood of the data. We're testing this on TCGA. So this is again bulk RNA-seq. And we have roughly 600,000 clinical variables. There's a lot of missingness in clinical variables. For instance, in some cancers, you measure one thing. In other cancers, you measure another thing. So it's a natural part of this type of data that there's a lot of missingness. So we thought it was a good example to use for our model. So here we can actually use, for instance, the mRNA to predict. So after training the model, we can predict the clinical variables from the mRNA or from a mixture of mRNA and the other variables. And we can impute missing values and so on. This shows a UMAP of the latent space. So you can see the model learns the different cancer types to separate them quite well. The colors are the 32 different cancers or whatever it is. And this is totally unsupervised, of course. This is like PCA. It doesn't know anything about cancer types. It has to learn from the data. Sometimes, of course, you see clusters that are pretty mixed up, but often we can explain that. So for instance, this cluster is all squamous cell carcinomas. I probably pronounced that wrong. So even if you have overlapping cancers here, it can often be explained by various subtypes of the cancers. Then we can predict from mRNA the clinical variables. I'll skip over this a little bit quickly, but it can reasonably well, for instance, predict age from mRNA expression. BMI not so well, but generally it's doing a reasonable job. And I mean, this is a model trained. It's a completely general model. It just models the output distribution of all these variables. But we can use it for prediction, for instance, also predict survival. So the model predicts the risk of death, and we can see that even though, again, this is a general model, when we compare survival predictions using the C-index that is used in survival, we do as well as models that are trained specifically to predict survival. So supervised learning models where you have all the other variables as input and predict directly the survival. And we can see these are survival curves calculated from the mean of the Gaussian representing the cluster for the different cancers. And this looks very much like what you expect for these different types of cancers. Again, we are obsessed with perturbations. What can we actually predict perturbations? And this plot just shows sort of the gradient flow in latent space. These plots are similar to PCA, but instead of using the first principal component, we actually use the mean direction of the gradient. They show gradients calculated on the predicted risk. So one of the output units give you predicted risk. We can calculate the gradient of the latent representation with respect to the risk. And then we can visualize those as these flows with arrows. So, and you can see that they go from low risk, yellow, to higher risk in these cancers. So actually we can now ask, how do we get this patient to have lower risk? We can move it along the gradient, and we can see what happens to the treatment type, the dose of whatever drug, or what happens to this or that gene. So hopefully we can use that to actually try to predict what to do. So now I'm going to a completely different type of multimodality. It's probably a stretch to call it that, namely having different types of latent representations. So we want to, for instance, disentangle disease from control. We would like to have a latent space for normal cells and sort of a parallel latent space for what happens when these cells have some sort of disease. So it's a very simple change of model. So this is the original model where we have this represents the vector corresponding to a latent value. Now we just add one more latent space. So this is sort of the baseline, normal, control. The normal latent space, then we add a conditional latent space that we call W here. This one we call Z. So we sort of extend the vector with an additional vector that models this. So we expand the input to the decoder in this way. Then we say that if it's a normal sample, it has to have W equal to 0. So we will have all zeros here if it's a normal sample. And if it's a disease sample, we learn the distribution in this space. So it means that a disease is sort of a combination of a normal cell and a disease. So we tried it on a data set, a single cell data set here, where we have 3 different infection stages. We have a salmonella infection and another infection measured at day 3 and day 10. And these are UMAPs of the latent spaces. This is the normal or the control. So the normal space, set space, and you can see if we color it with control or infected, they overlap completely. So you cannot distinguish normal and disease in this space. This is modeling the normal component. If we color by cell type, we see a pretty nice separation by cell type. But in this conditional latent space, you can see we learn the 3 different infection stages. So we have a very nice separation of infections in this. So remember, the normal samples are 0 over here, so we don't see them. These are only the infected cells. So we managed to disentangle these 2 or this disease effect quite nicely. How am I doing on time? Okay, but that's good because I'm more or less done. We actually use it also in the multi-DTD. We add another latent space to model batch effects. So instead of having just 1 latent space, we have a model of batches. So for instance, if you have 3 different labs doing experiments, we can have 3 different clusters here. And this can be used for removing batch effects. That's all I have to say. I just want to thank my group. And I'm part of the Center for Health Data Science, where we, by the way, teach a lot of our courses, including bioconductor. I am supported by the Novo Nordisk Foundation. Thank you.
Thank you very much for this interesting talk. Are there any questions? Maybe we start here.
Yes, thanks for a nice talk. My question is basically based on your model to predict micron expression from mRNA expression. So as I know, like the single cell microRNA sequencing data analysis or the technology is not well developed, and the drop-off events in the microRNA is quite higher than gene. So I'm wondering whether do you think like your model could predict a single cell microRNA expression based on this situation? Thank you.
We're trying to move in that direction, but you're absolutely right. We have very little single cell data to test on. So the hope is that we can use bulk models to maybe predict what happens in single cell. So we have some evidence that it's working, but it is difficult to test because of the lack of data. Yeah.
Hi, great talk. I was wondering if you could also easily get uncertainty estimates on the predictions and on counterfactuals that you're calculating.
Absolutely. So this is a fully probabilistic model. So we get for each variable, we get a probability distribution. So yes, we can get uncertainty.
I think there were more questions in the room. Yes.
Yeah, great talk. So just want to ask, like, you are trying to match your hidden space with output space. So in theory, like, you are trying to match two distributions. I mean, it's like, how do you actually do this kind of one-to-one mapping? So let's say I have a cell, I have the mRNA expression, and then I have output that will be the micron expression. But you are doing like predicting the bulk level, right? So I don't know. I think like it's probably not possible to have like a single cell expression and the micron expression in the same cell. I think your data is probably MR expression and then bulk micron expression.
It's all bulk.
It's
For
all
the
bulk.
microRNA, it's all bulk.
Okay.
So it's the same sample where they measure microRNA and mRNA in bulk.
Yeah. So the question is like, now we have a distribution of hidden space, and then we have a distribution of some abstract output space. So how do you do this kind of one-to-one mapping? I mean, if you don't have like data entities that are one-to-one.
But we do have one-to-one. So we have paired samples of microRNA and mRNA.
Okay. So your data set, the input data is one-to-one mapping.
Yes.
Okay.
Yes.
Okay. And another question is like, how do you design your hidden space? It's like a Gaussian mixture.
Yeah.
But then the data are usually not Gaussian.
No. So for the output distribution, we use a negative binomial if it's count data. So
yeah.
the output is a scaled mean of the negative binomial.
Yeah.
And we're also learning the dispersion or whatever.
Yeah, I mean, so on the left side that you have a hidden space as a Z, right?
Yeah.
So how do you define that? So you mean like you mentioned that Z is like a Gaussian mixture.
Yeah.
And so it's just how do you actually select the distribution? Because then you can just create
we learn the parameters.
You learn the parameter. Okay.
So it's part of the model. So we learn the means and the covariance matrices for the components and the mixture coefficient.
Okay, great. Yeah. Thanks a lot.
You're welcome.
Thank you. I'm sorry that I need to stop the questions here for the interest of time. Maybe let me quickly take the online question. Is it possible to perform in silico gene knockdown or overexpression as well?
Yes, absolutely. We can do that. But as I said, if you try to go from all the way sort of to the end state, it doesn't work. You can see the direction that the cell is going, but you cannot sort of predict the end state. The model is not capable of that.
Thank you very much. Let's give a hand again for our keynote.